Good morning and welcome to Plants, Pests and Pathogens, our first webinar in the 2025 season. I'm Charlotte Glenn. I'm the Extension Master Gardener Program Manager, and I am so excited to start things off today with a few opening announcements. Then I'll be turning it over to Matt Bertone and Mike Munster with the NC State Plant Disease and Insect Clinic, and they will be talking about scientific nomenclature, why it's important, why we all need to use it and be familiar with it. Later on, after our feature presentation, we'll have our Be on the Lookout section, which is something we do every Plants, Pests, and Pathogens, where Matt and Mike share insect pests and plant diseases that we should all be on the lookout for in the coming weeks that we're likely to see out and about. And I'll finish things up today with a new section that we're going to have in each of our webinars called Plant These for Pollinators, and then some Extension Master Gardener program announcements. So our team, you might notice, is a little smaller this year. Caroline Richardson, who was just wonderful program assistant with the Extension Master Gardener program, she decided to move on to other opportunities at the end of the year. So this year it's going to be myself, Charlotte Glenn, with the NC State Extension Master Gardener program, and of course our dynamic duo, Matt Bertone, who is the director and entomologist of NC State Plant Disease Insect Clinic, and Mike Munster, plant disease diagnostician. Both Matt and Mike are based in the Department of Entomology and Plant Pathology, and we will be with you this season, bringing you lots of information about sustainable gardening tips and also diagnostic tips, so you can learn to recognize plant diseases and pests, because that will give you the information you need to then find information about research-based recommendations and to learn more about those issues. So that's just what this slide is telling us with Plants, Pests, and Pathogens is the fourth Tuesday of each month in the growing season. So in North Carolina, we count that as from February to October. We always start at 10 o'clock and run till 11:30. Of course, this is a continuing education webinar series. The live webinars are open to our Extension Master Gardener volunteers and extension professionals. And again, our focus is on those diagnostic tips and sustainable gardening topics. We don't go into a lot of recommendations on control for the pests and diseases that are discussed, because our goal is to give you the information you need to then find those recommendations and additional information by helping you identify those problems. So we have in every one of our webinars, of course, will count towards your EMG certification requirement, the continuing education hours part of your annual requirement. So be sure to sign up on the EMG intranet. That'll create a continuing education hours submission for you, and then you'll just be able to submit it and keep up with your continuing education requirement. And then each of our webinars will have a keynote presentation with one of our experts at NC State or potentially further afield, discussing timely topics related to sustainable gardening and integrated pest management. As mentioned already, like today, we'll have our Be on the Lookout, or BOLOs, those common pests and diseases you should see in the coming months. We're also going to be featuring our Plant These for Pollinators throughout the year, and we encourage everybody to stay on as we wrap up at the end with Extension Master Gardener program announcements, interest to our volunteers across the state. So you've probably already seen the schedule for this year, but just to review it quickly, we will be continuing our pattern of having Matt and Mike be our feature speakers every other month. Starting in February, today's webinar is going to delve into the topic of scientific names. In April, they'll be back to talk about, well, they're here for every webinar, but they'll be our feature speakers again in April to talk about pests and disease of berry crops, including blueberries, strawberries, raspberries, and blackberries. In June, we'll be taking a dive into the pests and diseases associated with weather-induced stress, whether it's drought or flooding or other weather-related issues that cause our plants to become more susceptible to problems. In August, we always have fun in August. They'll lead us through diagnosing pests and plant diseases from photos that you submit. So be thinking about that through the year and collecting photos of unusual insects or symptoms that you're seeing on your plant. So we have those ready for our August webinar. And then in our October wrap up, we will have Critter or Not, which is always extremely fun, educational, and very popular. And Matt and Mike do an excellent job of showing us some of the more unusual or puzzling issues that have come into the plant disease and insect clinic that we can all learn from. In the other opposite months, when Matt and Mike aren't our feature speakers, we have guest presenters that will talk on a variety of topics this year. We're excited to have next month in March, Amy Bruzacchese, who is our therapeutic horticulture program manager, talking about understanding gardening groups and therapeutic horticulture. In May, Debbie Roos, who's the extension agent in Chatham County for sustainable agriculture, will be telling us all about allium leaf miner, which is a new pest in North Carolina, and what we need to know about it. In July, Lucy Bradley, our consumer horticulture specialist, will be sharing climate resilient strategies for home gardens and landscapes. And then in September, we'll welcome Michael Reiskind with the NC State Department of Entomology and Plant Pathology, who's going to talk about smart mosquito control. So what we can do to manage mosquito populations, but also protect pollinators. So the full schedule is online and you can check that out. It's also, all of these are posted on the EMG intranet calendar, so you can sign up for them now. Quick reminder where you can find our resources for plants, pests, and pathogens. There is a page on the North Carolina Extension Master Gardener intranet. So you'll have to navigate from your county EMG intranet to the North Carolina site. We've even created a Go link that you can bookmark, so you always have easy access to it. But this is where you'll find the schedule, the registration information, and the recordings. So from your county site, when you first log into the EMG intranet, where you open on your county site, to get to the North Carolina site, just click on the North Carolina link, which is up at the top, right under the And just a reminder for folks to keep their mics muted. So once you're on the North Carolina site, check out the red column that's on the left-hand side and go down to where you see plants, pests, and pathogens. It is one of the topics there. And when you click on that, that'll open the plants, pests, and pathogens page with more information about the webinar series, a link to the schedule, and also a link to recordings. So this is where you'll be able to find recordings. On that page, there's a link that's to the 2025 season, as well as to all other seasons. So the quick link will take you to this current season, but if you want to see other seasons beyond 2025, click on that all seasons recordings and resources. See, we got a typo there, so I need to replace that S with an R. The other place you can find our recordings is on our YouTube channel, the NC State Extension Master Gardener channel. We have a playlist for plants, pests, and pathogens, so we've got a quick link for that too. When you go there, you will find almost all of our recordings up to the 2024 season. We aren't able to make these public on the playlist until all of the subtitles have been corrected. And with many thanks to a very diligent group of volunteers, we got almost all of our subtitles updated and corrected. And the last few are being worked on right now. So I want to say a really big thank you to our team who's been working on those all throughout the year, all throughout the winter, to make those available, so they're not just available to our Master Gardener volunteers and extension professionals, but to the wider public, so everybody can learn from them. So shout out to Don Burnell, Kathy Foster, Tammy Huey, Deb Young, Greg Shelton, Alicia Thorpe, Kim Wallace, Pat Welfare, and Kathy Williams. So those are our folks who've been working this past season, and to everybody, because we know we've had many volunteers over the years help with these subtitles. So thank you to everybody who's gotten us to where we are. We're finally almost up to date. This is the most up to date we've ever been with our subtitle backlog. So really appreciate the work they're doing to make these recordings available. So that is all of our opening information for today, and I will turn it over to Matt Bertone and Mike Munster for their feature presentation on adventures in scientific nomenclature. I will stop my share so you guys can start your share.
Great. Thanks, Charlotte. Thanks for all the work you do, and we're sad to see Caroline go for but we're really thankful for all the work she did in the past. And she was a great member of the team. Um, all right, so we are going to be talking about something everybody's gonna love. And we did get a lot of comments about, using scientific names, things like that. And unfortunately, it's unavoidable. But we're here to tell you what's going on with scientific names, what the deal is, and all this other stuff. So I'm going to run the show here while and Mike is going to chime in, and we're going to kind of go through this talk. And hopefully, at the end, you're going to know a lot more about why we name things. So first.
So we talk about the title of the presentation, of course, has to do with nomenclature, but we better define that before we start. Basically, nomen, of course, means name, and nomenclature is the assigning of names to different units, whereas taxonomy is the classification of those units. For example, if I say, here's Devon, here's Will, here's Jose, here's Maria, that's nomenclature. I'm giving names. But if I say Devon and Will are both Smith and Jose and Maria are both Villegas, now I'm starting to classify them into higher groups, in this case into families. So we will inevitably have to talk a little bit about taxonomy or classification, but our focus in the presentation is going to be nomenclature or the names themselves.
All right, and so with that, we're gonna start with this. So Charlotte, if you want to bring up the poll. So what is this? What is this? Anybody know what this thing is? And so this is Charlotte, the special poll I asked you to make. All right, so what is this? Oh, I see that has a yeah, so and it is not sorry, it's not beetle hoist. No worries, Charlotte. I understand.
Is this on over there? Is my speaker on?
When you type in the options, it doesn't automatically take out choice one, choice two. I must not have
Totally
noticed
understand. Well, it's hey, you
choice
can call
two.
anybody can call it that. I'm gonna start calling that Lamperiod beetle hoist. So
There you
yeah,
go.
so all right, so we're gonna give it another 10 seconds or so. Everybody wanna chime in? Okay. So I guess we can close it here in the interest of time. So we're gonna end it and then share the results. So a lot of people call it lightning bug. Some people call it firefly. Some people call it Lampyrid beetle. Lucianaga is another option. And Wawa tessi is another option. And here's the thing. All of you are correct because all of those are valid common names for this creature. In fact, when I was growing up in Pennsylvania, I called it a lightning bug, but now I call it more a firefly. Kind of, it's a little bit shorter and things like that. But yes, so this is a common name. These are all correct. And basically, things have names. And I know this seems obvious, but it's a way to communicate about entities. And so the language and culture you're in often dictate what something is called, but unfortunately, that leads to regional differences that can cause confusion and inhibit communication. So if you say, I found a Lucianaga, and somebody says, oh, I found, but no, that's a firefly. Well, you're talking about the same thing. So we really have to coordinate these things. And these things that we're talking about right now are called common names. They are, or vernacular names, colloquial names, things that people generally call these things, but can have often a regional difference. So for instance, I get somebody sending me a message, I've got water bugs all over my house. And I kind of know what they're talking about. But if you were to take that literally, there are lots of aquatic hemiptera, aquatic true bugs like giant water bugs and water scorpions, things like that. But I know that they're not talking about water bugs like true hemiptera. But this is a colloquial name often given for cockroaches. But furthermore, which cockroach are you talking about? I don't know if you're talking about the American cockroach on the left or the smoky brown cockroach or palmetto bug on the right. And in fact, you see there too that on the right, this entity, this organism has two common names. And if you say, oh, I'm talking about palmetto bugs, I'm like, oh, yeah, smoky brown cockroaches. There could be a big argument between that. And so it's really important to know that there are different common names for things. let's see.
Here's an example from the world of plant pathology where the word blue mold could refer to two very different things, a disease of tobacco or a disease of daffodil bulbs, one caused by a fungus-like organism and the other caused by a true fungus. And we have many such examples. For example, black rot means different things if it's on apple, if it's on grape, if it's on other different hosts. So common names can get us into trouble.
And even not only just different organisms have different common names, but even the different life stages in organisms have different names. So, for instance, the corn rootworm is below, right? That's a larva of a beetle. It's not actually even a worm, and so that's also a misnomer. But the adult form is called the spotted cucumber beetle. So again, you could be confused if you're saying, "Oh, I'm talking about spotted cucumber beetle," and this person say, "No, I'm talking about the corn rootworm." Well, they are both the same thing. Really important to know. So scientific names were developed. They're meant to standardize what we call organisms across languages and cultures. And there were various versions attempted historically, but the modern system we use was developed in 1758. Actually, it was the 10th edition of this book series, Systema Naturae, that Carl von Linné, or Linnaeus, wrote. And it wasn't until the 10th edition that he formalized this binomial. So each name consists of two parts. And this is a really important document. This is the most important document in scientific nomenclature that exists. And in fact, Linnaeus was actually a botanist, although in this book he described lots of different organisms. One of them happens to be Musca domestica. So here is a scientific name and the components of it. This is kind of the full scientific name, although I will say that we didn't mention this, but oftentimes the year is at the end as well. So when this was made, this description was made. But here are the components. So basically you have a genus, you have the specific epithet, and the author or authority. So Linnaeus described a type of thing in the genus Musca and the species Domestica. And when we actually talk about a species, we're talking about this whole combination, Musca domestica. Now, I'm not sure anybody knows what this actually is, but just in case you want to know, Musca domestica is the housefly. And so Linnaeus actually named this in that book, Musca domestica. And in fact, a lot of flies he named were named Musca. And nowadays, though, we include a lot of several species in this genus. So it's not just Musca domestica that's in here, but another species, for instance, Musca autumnalis, the face fly, was described by de Geer years later. And so even though they're the same genus, they're different species. But when we talk about the species, we don't just say autumnalis or domestica. We have to say Musca domestica because we need to know that this species epithet is in this genus. So we have a full kind of species name. And just to show, kind of highlight why, these species names, this type of binomial transcends language. Here's a Chinese manuscript, and you can see I know what this is speaking about because I know what Aphis glycinis is the soybean aphid. And because even though there's all these Chinese characters in here, I know that this is about this because this transcends languages and cultures.
One of the other things to mention here is as kind of analogous to Matt's slide of the corn rootworm and the cucumber beetle being different life stages of the same organism. With fungi, many of them have two different stages and very different morphologies between those two stages of formation of spores. One we call the teleomorph, which is the formation of the sexual stage, and the other called the anamorph, or the asexual stage. Now, I don't know if any entomologist was so foolish having thought that the cucumber beetle and the corn rootworm were actually two different organisms, but these fungi, when they're only encountered in one or the other of their morphs, may have actually have been given different names. So we had for a long time a whole set of parallel names, depending on if you were talking about the sexual or asexual state of reproduction of the fungus. Then a rule went into effect in 2013. Wow, it's been a long time now. That was called the one fungus, one name rule. And that specified that you can only have one valid name for the fungus, not two anymore. This, of course, caused some consternation and some adjustments. And there are different, because of the rules, you can't necessarily predict which of the names is going to be the actual accepted name. Now it has to do with things like priority, you know, which one was first and so on. And two helpful sites, and we'll show a screenshot from one of these later, are mycobank.org and indexfungorum.org for more information about fungal names, which we'll mention later.
Yeah, and just to follow up on that, Mike, yeah, there were in the past, say, people that would describe a female versus a male of the same species as different species if they're very different. In fact, Francis Walker, who has described 20,000 species, many of them were really bad or just different color morphs or things like that. And that's a whole other topic we could talk about. But okay, so where do these things fit? So basically, all of these names fit in a higher level classification. So, for instance, we've got two examples here. We've got an insect and a fungus. So both are cellular organisms. Both are in the superkingdom Eukaryota, which means they are not bacteria or prokaryotes. They're not viruses either, but they have true eukaryotic cells. But here's where it differs then. So the insects are in the kingdom Animalia, fungi are obviously in the kingdom Fungi. The insects are arthropods. This fungus right here is a Basidiomycota. Well, Mike can maybe you go down that side. But as yeah, go ahead, Mike.
Yeah, no, Basidiomycota would be the phylum, but the common name for that, because some of the higher taxa also have common names, is the Basidiomycetes. So you may see that with, of course, a lowercase b.
Yes, and that's another thing. Yeah, lowercase versus uppercase. This beetle is an insect. It's in the order Coleoptera, which is all the beetles. It's in the family Colonariidae, which are the turtle beetles, and this is called a turtle beetle, common name. And the genus Culinarium and the species Culinarium lecanti, and it was named by Thomas. And then Mike, if you want to describe the.
Sure. So we part ways here at after the super kingdom. So the kingdom fungi, although actually fairly closely related to the animals, is separate from them. And then the phylum Basidiomycota would be where this and most other macrofungi belong, Agaricomycetes and Agaricales. So one of the things you would see there would be those that have gills on the underside. The family Phycelaceae, genus Des Armillaria, and I'll talk a little bit about, think later about the whole why names change, because you may have known this fungus as Armillaria tibescens, but now it's Des Armillaria cespitosa. And is it here or later we're going to talk about the parentheses and the authorities?
Yep, we'll talk about that in a second. Yeah.
All right, so we'll leave you to wonder at the runes following the name there. But when Matt put me on the spot is what the common name is, I said I think it's ringless honey mushroom, but we'll go with that.
Yeah, and again, common names are not actually that common. A lot of things are only known by their scientific names. And so every organism can kind of climb up this tree, and the higher levels change. We'll talk about a little bit about that later on. But one thing I did want to point out was that you can see that certain things, certain levels, especially family level, they actually have consistent endings. So you can actually tell something from that word. So if you see any kind of word with an IDAE and it's an animal, we know that it's a family of animals. So for instance, Aphididae are the aphids. Culinariidae are the turtle beetles. And plants and fungi always have ACA as the ending for their families. So Lauraceae is the laurels and things like that, and whatnot. So just some kind of little hints at what taxonomic level you're looking at when you see that ending. Oh.
And I wanted to add there too that if you get above to where we had, for example, phylum and class, you notice the ending there, Mycota and Mycetes. So if it were plants, even though they're under the same code, which we'll see in a minute, that myco there, which comes from the Greek word for fungus, is going to be the tip-off that you are looking at a fungus and not a plant.
Okay. So codes of nomenclature. So basically, who governs all this? And really, there's been over the years, there are different commissions of officials that write these codes, and kind of it's a rule book on how we name these things because we do need to standardize these things. And so, for instance, the ICZN, which also the commission is called the ICZN, but the International Code of Zoological Nomenclature, it governs living and extinct animals, protozoa. There's the International Code of Nomenclature for Algae, Fungi, and Plants, formerly the International Code of Botanical Nomenclature, International Code of Nomenclature of Prokaryotes, so that's bacterial names, and then the International Committee on Taxonomy of Viruses, which Mike, when I'm done, if you want to give a little hint about that, why that's this asterisk after it. But one of the points I want to show here is that each code, technically within a code, like all animals have to have unique names. So there cannot be another animal at all, extinct or living, that is named Pieris. But because the different codes apply to different groups of organisms, technically this butterfly is Pieris, but this Andromeda is also Pieris, the genus Pieris. So if you see a plant and an insect with the same genus name, that's okay. You can't have the same plants have the same genus name and animals and whatnot.
So the virus thing, yeah, the I guess there's a lot of asterisks there. One of them is probably indicating that I am a little bit ignorant about it. Another one is that things seem to be in flux. So we had commonly used English names as the scientific names of viruses. You're probably familiar with things like tobacco mosaic virus, tomato spotted wilt virus, and so on. But there's a transition now toward using a binomial system. So stay tuned on that. We won't really be delving into that.
All right, so formatting scientific names. So when you see a scientific name, you italicize the genus and specific epithet, or if you're writing it, you can underline it. Handwritten or typewriters used to use that as their way to notify that you have got a scientific name, and you'll see that throughout here. The genus can be abbreviated after the first use in the same document. So Musca domestica, if you're writing a paper about it, you can just reduce it to M domestica. T-Rex, as you'll see in a second. But special attention when different names begin with the same letters. So Phytophthora infestans and Pythium irregulari both start with P, so we add an extra letter just to make sure that we can tell. But this saves room when you're writing out papers and whatnot. So if the author is in parentheses at the end of it, it was in a different genus than originally described. So if Musca domestica was now put in a genus Tipularia or whatever, some crazy thing, Linnaeus's name would be in parentheses then to show that he described the original species, but that it's not in the original one that he described it in the genus. For plants or fungi, most recent authors also follow, and we'll see that in a few examples coming up. Some author names are abbreviated. So, for example, in animals, Linnaeus and Fabricius are written L and F because they're kind of one of the, they're way back when old school kind of originators of a lot of this stuff. And if you want to generalize things, you can reduce the name species to abbreviation. So SP is a single species, SPP is multiple species. So, for example, Xylaria, a Xylaria species is an unknown or unidentified species in the genus Xylaria, whereas Xylaria SPP means multiple Xylaria species. So if you're talking about all the Xylaria species in North Carolina.
So as Matt was saying, there is a difference between how we handle things with fungi and how the entomologists do with arthropods. Here is a powdery mildew called Potosphaera alpina, and the S. bloomer would have been the person who originally named the fungus, but then when it was renamed or moved into a different genus by Brown and Takamatsu, I think was his name. These we do abbreviate more. Then they get their names appended to the scientific name, but retaining always the original describer in parentheses so that you've got a link back to what's called the basionym, or the original name that the organism was given. As far as pronunciation, there is in a book that I've got, Botanical Latin, a wonderful statement that the pronunciation, the important thing is that it sounds good and everyone who hears you can understand what you're talking about. So this is a written scientific names are a written language and not a spoken one. So is this Kalanchoe or Kalanchoe? Is this Forsythia or Forsythia? As long as we understand one another, we're okay. So not to sweat about that. If you do want to sweat about it, I think there was rats. I think it was in the University of Wisconsin, and a key to the Asteraceae, they go a little bit more into the pronunciation of things. So there are potentially resources out there if you want some tips.
Yes, and just saying it with confidence is the best thing, but it's and I talked to Mike about this. I took Latin. I think he took Latin too in school because we like this scientific stuff, these names and things. And what we can discern from the spoken language of Latin is actually fairly different. There are a lot of things that just aren't present in the English language, or vice versa. So just do your best. This is, like Mike said, it meant to be a written thing. And this moves on to things that we take for granted that we actually say the scientific name. So if you've said Tyrannosaurus Rex ever in your life, you have said a scientific name, and it's very commonly known, very easily known. And in fact, most dinosaurs, just about all of them, you're talking about most often their genus. Like Stegosaurus is a genus, Triceratops is a genus. So you already use them more than you think.
And one of the questions that was in the chat earlier was about whether they're all in English. Well, they are, of course, in the alphabet, the same alphabet that we use, but the roots tend to be based in either Latin or Greek. Now, not 100%, but that's going to be your most two common languages of origin of these names. And so if you know a little bit about that, and yes, here is a good example, Trifolium repens. So tri, of course, three, folium, leaf, and repens means creeping. So a creeping three-leaf thing would be the scientific name for what we in English call white clover. Here's an example from the fungal world, Botrytis cinerea, the botry part. Anytime you see that, you're talking about something that's going to have clusters in some way or another. So it comes from the word meaning a cluster of grapes, and you can see that the spores are reminiscent of that when you look at them under the microscope. Cinerea, and a lot of these specific epithets actually have to do with colors. Cinerea means ash gray, which is the color macroscopically when you see the fungus sporulating on the surface of a plant.
Now on from the insect world, so Necrobia rufipes. This is one we call generally the red-legged hand beetle, but the genus name basically refers to the death lifestyle. This is because this beetle feeds on dead, dried meat, dried carrion. And then very common in insects and in animals, rufipes literally just means red foot. And so anything with red legs often has the name rufipes.
And another common source of specific epithets is the host when you're dealing with pathogens. So in this case, Diplocarpon rosei refers to the fungus that causes black spot on rose, and the rosei, of course, indicating what the host of this fungus is.
So that's where we get to things like this, Triaza magnoliae. And this is Ashmead described this in 1881. What plant is this? Does anybody know? Anybody want to guess?
Magnolia?
Yeah, seems like it. But no, it's red bay, as somebody said. So what happened was the person who described it misidentified the host. And so that name will persist forever because it's valid and it is the oldest name for this entity, this insect. But it will confuse people forever too, because it's not on magnolia, it's only on red bays or laurels. So Now, be careful, especially also when you say, when they have locations. So like Illinoisensis or something, that does not mean it's necessarily only from Illinois. So be very careful sometimes when translating them literally because there might be mistakes.
And we got a lot of Caroliniana too here.
Yep.
Just Matt, so this critter is causing that galling on the edges of the leaves.
Yep. So yep, it's found in there. And if you're on the coast, you'll see this fairly commonly. This not the magnolia psyllid, but the red bay psyllid. And of course, scientific names can be come from people's names if they're named after that person. So, for instance, Calanectria hodgesii is a fungus that was named after our late friend and diagnostician, Dr. Chuck Hodges, in the lab. So you can name things. People can have things named after them by other people. It is kind of a faux pas to name something after yourself, but you can name it after your spouse or your cousin or whatever. And then if some people really respect you, they'll name it after you or if you collected it. And just to give another highlight, another person in our lab, Dave Steffen, the late Dave Steffen, he has a tree hopper named after him, Telamona steffeni. And here's where to just show this is actually based in Latin. So these are called patronyms, even though it's not just naming after men. But basically, the ending, when you see that name, Stefan is his last name. You add the I, that's a masculine possessive in Latin, means Stefan's Telamona. If you were naming it after a woman, it would be AE, so it's this person's and then the feminine. Or you can technically even name it after all of Dave's family. So Stephanorum would be all of the Stephans. So it's a plural possessive. So this is kind of the rationale of where these things come from. They're basically possessive, and that's also what the rosy was when Mike talked about that fungus. It's of the rose. And me and Mike refer to this book I have in the lab, Composition of Scientific Words. It's really fun to be able to look up. You can look up either the English terms and see what the different Greek and Latin words are, or you can go from the Latin to the English. And so, for instance, nathos, jaw, that's a very common one. Pronathus, for instance, is insects with jaws that are facing forward. And pro meaning forward. So this is where a lot of scientific names come, or even terms come from.
Yeah, I told Matt one time that if this book ever disappears from his shelf, I need to be suspect number one.
It's a great book. We've got to get you a copy too, but it's so much fun to see these things. I mean, maybe only if you're geeks like us, but anyway. All right. So what is a species? It seems obvious, but is very difficult to define. And this is where we get into, okay, what is that entity? Like you have a name, but what is the actual thing? So oftentimes biological species concept is most common. So things that interbreed are species, but there's a lot of issues with that. So asexual animals, talk about hybrids in a second. And one thing to note is that there are type specimens of each species out there. So there's a specimen of every plant, of every fungus, of every animal that is in a collection somewhere that hopefully some are lost, some are whatever, borrowed and never come back. But they sit in a collection and are the representative of that species. So if somebody wants to know whether the specimen they have is that species, they can go back and look at it and say, oh yeah, this matches exactly. This is what it is. But there's a lot of other things going on with species.
Sometimes a species isn't specific enough, and we want to make further refinements in how we classify things below the species level. For example, the soft rot bacterium that some of you may know as Erwinia caratava is now called Pectobacterium carataverum. Notice how the ending changed though to match the gender of the noun there. The subspecies, there are a couple of subspecies, one of which is also called carataverum. So that is one way that we would divide it. It tends to be more in bacteria that we would see that, but also, and I'll let Matt comment about the animals.
Yes, so subspecies are not super common in animals, but they are dictated by the code and they are accepted. So, for instance, that corn rootworm or the spotted cucumber beetle here in the east is Diabrotica undecipunctata howardi. And in the west, there's a western version of it that's a different subspecies. So, and again, it really depends on the taxonomist to say whether these are true species or not. But if they're if they can interbreed, but they've seemed distinct enough, maybe you put a subspecies on them.
And in reference to how they interact with plants, there are designations called forma specialis, races, and pathivars. So this would be more common than seeing a subspecies, which really is also not that common for the pathogens. But forma specialis are. So Carnartium quercum forma specialis fusiforme is a division within the species Carnartium quercum, and that is the one that designates our fusiform rust, the fungus that causes that. Xanthomonas hortorum pathivar pelargonii. The pathivar refers to the variety that causes pathogen, or excuse me, is pathogenic on, in this case, pelargonium. So that is the bacterial blight of the, well, common name geranium, scientific name pelargonium. If there are if there are genes for resistance that have been used in plants to try and help them survive, prosper, produce in the presence of a pathogen, sometimes that, well, that will cause, of course, selective pressure, and eventually we'll get pathogens that overcome those genes for resistance. And then sometimes we will designate those as races. Now that should not be a capital C there. I think the autocorrect wanted to do that because that C should be lowercase. But Fusarium oxysporum forma specialis cubense, which would designate the banana affecting forma specialis of the fungus Fusarium oxysporum. It's a vascular wilt pathogen. Tropical race 4. So that would be one that's of a great deal of concern right now in the banana growing world, is causing a lot of destruction. So technically there are some incorrect things about the way that this is laid out. The FSP should not be italicized. The race 4 should also not be italicized. But just an example of a division within a species that we sometimes use in pathology.
But you put that in there on purpose so people would know and they would see that immediately and know, right?
Yeah, I didn't want people to feel bad, so I didn't confess that. Yeah.
All right. And then,
so with plants especially, although it can happen with other organisms, we have a lot of hybrids, and those can be designated by a multiplication sign. You can either have it between the two full names of the elements that went into the hybrid, or you can give it its own name. And here are two examples of that. So an interspecific hybrid would be one within of two different species within the same genus. So our foster holly is Ilex attenuata, and there is that, it's actually a multiplication sign, although you can use an X if you don't have a multiplication sign. It's associated with, but not part of, the epithet in this case. If it's an intergeneric hybrid, so two different genera were crossed to be able to get the plant, then that multiplication sign goes out in front of the genus. And our very common Leyland cypress is a case in point. The Hesperotropis leylandii, it is now called. But that multiplication sign does not, for example, enter into the alphabetization of the name. So don't go looking it up under X.
do you want to talk about this, Mike?
So yeah, here's where we have to kind of show a little humility, because even though these scientific names were supposed to be standardized so people understand them everywhere, and we don't have to worry about language and regional variation in the common names, even so, sometimes the scientific names, excuse me, do change. But there are valid reasons for that. Sometimes it's because there is a species complex, or what I'll call in another slide, cryptic species, where folks realize that, oh, this is not just one thing, but there are actually two different species here we've been calling the same things, and then we have to name one differently. It could also come from a new understanding of the relationships. So maybe we say that we've been grouping these species in the same genus, but there are reasons to say that they're not all that closely related, maybe from, or very often nowadays, of course, from genetic studies, and we decide that it merits moving some of them into a different genus. They don't have as close a relationship as you would have thought originally. Sometimes also it's a case, and I'll show an example in a little bit here, where the understanding of a species or name that was applied, let's say here in North America, was based on a species concept from Europe. This happens with mushrooms, for example. And later on, folks find out that no, what we've been calling here in North America, let's say Heterobasidium annosum, previously Phomizinosis, no, it's not the same thing as that. We have to come up with another name, in this case, Heterobasidium irregulare. And I think I mentioned the armillary one before, but I may come back to that one. So there are reasons why the scientific names will change. There are also reasons based on the rules. So priority is very important. So who named it validly, a validly published name first? That will decide what the correct name is, or at least part of the rules that determine that. There are other things that happen, like for folks who've been around, we remember the grass family once called Gramineae, and the brassicas once called cruciferae. But now those names have changed because of the rule that the family name needs to be based on the name of a genus, which needs to be based on the name of a species, the type species in that. So there are some name changes that have been based on the codes themselves.
Yeah, and you all can see that the POAC ends in that ACEA AC, and that's based on POA, is the genus of grass that it's based on. So that's the type genus, the genus of all genera of grasses, basically the original one that was described as a true grass. So yeah, and this is why things change, unfortunately. Now it is frustrating even for taxonomists and people like us, and we do have to learn new things every once in a while. But if they stabilize, then it becomes kind of common knowledge again.
So here's an example of that. A fungus that we used to see and confidently identify, I'll give you the name in a moment, turned out to be more than one species actually when they really dug into it. And then we also have the problem of synonymy, or names, two names for the same thing, but of course only one of them is going to be the currently accepted name. So if you go to the next slide, Matt, these are spores, Alluria canidia, of the fungus that causes black root rot on pansies, on Japanese holly, on a lot of herbaceous perennials. But it turns out that they decided that it doesn't belong with the other members of the genus Thalaviopsis, and they discovered that there are two species, not one here. So now the names are Berkeliomyces basicula and Berkeliomyces ruxiae. And one of the things that I love about that name is you'll notice that A, E, I, O, and U are all present in that specific epithet. There's another one that's like that too, sequoia. So some just little fun things that we run across in the labyrinth of these names sometimes. All right.
All right. Let's see. And sometimes these taxonomic changes become a problem. So for instance, there is a genus of flies you all probably know. You probably heard of the genus Drosophila, and that's again a genus that you all pronounce and you all say. So, well, maybe not all of you, but you know this thing called Drosophila. Well, unfortunately, a lot of genera were at one point, based on molecular data, were elevated. And what happens is the Drosophila melanogaster, the kind of really commonly used genetic model organism that's been used in science for decades now, would be then Sophophora melanogaster. And this would have changed so much, and so much, it would be very difficult then to look at old literature, things like that. So what people did was to petition the commissions to conserve the usage of these things. So they basically say this is such an important species name that we can't just go by the rules. We'd like to petition for that to be kept. And there's all this kind of legalese stuff and all these kind of descriptions of what they're proposing and solving the problem. It is in the weeds. It's not that we're going to discuss specifically, but sometimes these things, even if they don't go against the rule, are conserved just because they're very functional or a very famous species, and we don't really want to get rid of that name. So you might be asking, okay, why does this all matter? And you might be asking practically, what does it matter to me? So this is really important because the correct name unlocks the associated information. So if you're saying, I want to know about midges over here on the right, there are 4 different types of flies here that you could technically call midges. But which one are you looking for? Which one do you need to know about? Because some are biting, some are not, some are plant feeders, some are things like that. And we just talked about earlier, common names have obvious flaws. They're regional. Different people know things by different things. And so searching literature is more specific using scientific names as they are unique. Of course, that does change, like we said. So sometimes I do have to search older names because I know that the literature is going to have the older names. And so that's where the difficulties do arise when the taxonomy is confused or changes a lot. One really difficult thing, and this is kind of what Mike, kind of in the situation with the black root rot that Mike just mentioned, is, for instance, you have one species and now it's split into 2. If you're looking at the old literature, which species are they talking about when they talk about the biologies or these older information? So when that happens, it's really difficult because unless you have voucher specimens where somebody placed those specimens in collections, you can go back and say, oh, this study looked at this species. It wasn't the original species. If those aren't present, then we really don't know what people are talking about. So there's not a silver bullet against confusion. But I will show that, for instance, if you just search biology of skippers, and I'm looking for skipper butterflies, I search on Google Scholar, and you're going to get all these things about fish called skippers, mud skippers and things like that. And I'm like, oh man, I got to wade through all these fish articles just to find the butterfly ones. But if I say biology of Hesperiidae, which is the family of skipper butterflies, then I get much more specific results to all the skippers, the butterflies that I actually want to know about. And if you're wondering where to find all this information, catalogs in animals are really great. I used to not like them because they don't really help with ID, and so for me that wasn't very helpful. But I will say now catalogs are really amazing in that they basically list all of the previous synonyms, information about where they are, some remarks about the taxonomy. So you can go in and say, okay, the recognized taxon here of this Scarabeetle is Echinatha castanea, and Erickson has the original one. But you can see that Erickson, it was originally described, it was actually described as that, but different people put them in different genera, and it came back to this one. So we have the true name currently, but we can see all of the history of where the names were, who made those decisions, what publications, and whatnot.
And just an example, we wouldn't call it a catalog necessarily, but in the fungal world, and if you go to indexfungorum.org and do a search, and then when the name comes up, click on the name and you go to species fungorum. So this is our Des armillaria cespitosa. And you can see that, in fact, yes, it was at one time called these other things. Unfortunately, this doesn't show you the link to it having been called Armillaria tibescens at one time, but that's again because oh rats, what is the name of a group, an organism that's similar to one in a different geographic location? There's a wonderful name that I can't remember right now. But so it turns out that Des armillaria tibescens, from the Armillaria tibescens, is a European species, and the one here is Des armillaria cespitosa. No, it's not ecotype. It's
Yeah, and it's not congenitor, because congenitor is just
yes,
anything where
yeah,
it's
I think that may be it.
Okay. Well, congenitor, I think of anything that's in the same genus as it.
No, then
what
it's,
you're
then it's
talking
something
about.
else. Anyway,
Okay.
so go ahead, Matt. Yeah, I'm sorry.
No, no worries.
You know, by the way, we are creating a great headache for the folks who are doing the subtitles on this particular
Oh my gosh.
episode.
Um, so yeah, so that's how, those are kind of two ways we can look for this history, and we can find out, okay, is the thing we're talking about a different thing, or is it just changed names or whatnot? And again, a lot of this comes because we're trying to refine more and more the actual entity and the definition of that entity, be able to unlock that information. So, finally, one thing, why else is it useful? Well, it literally saved Luttrell's life. And Luttrell was this biologist back in the old days, and he was imprisoned. And while in prison, a physician was checking out the prisoners and came up to him, and Luttrell noted that he saw this beetle, and he identified it to species. And the physician was so impressed with that, he understood that this wasn't just any ordinary prisoner, and petitioned to get him released. And this is a very famous taxonomist, especially in arthropods, but also lots of other life. And so I guess I don't know if that's going to help you practically day to day, but just a kind of a neat story about how taxonomy has saved at least one life in history, and probably more, depending. Okay, Mike, you have anything to add to that?
No, I do not.
Okay. Well, we're gonna wrap up with the final question, and then this will be an A through E poll, Charlotte. But yeah, so we really appreciate you listening to this, and we're gonna answer some questions in the chat. But here is let me go to the next page. Okay, here's your quiz. So, what's wrong with this scientific name? And now you all should be able to tell us. All right, we're gonna give everybody a good amount of time to take a look. Now, this happens very commonly in media when people are not science writers or don't encounter scientific names very often. And they'll just kind of write it like you think you would. And unfortunately, as we've seen, there are rules to this, and it's really important so that we can identify scientific names among other things. All right, we'll give this another 20 seconds or so. People in the chats and stuff. Okay.
I don't have any choices. I just have the letters.
No worries. Yeah, I don't know, on some devices,
It's not on the survey, it's on the screen. The choices are on the screen.
yes. So I can just tell you right now real quickly, A, it's not capitalized. B, it's not bolded. C, it's not italicized. D, nothing is wrong. Or E, both A and C. All right, we will end it there for this poll and see how we did. So looks like the correct answer is E, both A and C. So it should be italicized, and the genus name should be capitalized. Nothing else should be capitalized. So the H in the Hyalinobatrachium, see, even it's difficult for us to pronounce. Hyalinobatrachium, which means glass frog. It should be capitalized. So E and A and C. So bolding doesn't matter. Bolding can happen. It's not against the rules. But you do need to have the whole thing italicized or underlined. You have to have a capital genus and a lowercase species specific epithet. So that's it for what we want to present today. I know people are really it's kind of daunting and a little kind of scary to talk about scientific names, but again, you know, it's really much better to be able to say which rose are you talking about? Rose of Sharon, which is a a hibiscus, which is technically the Malvaceae, the cotton family, or a rose, which is in the Rosaceae, which includes things like strawberries and apples and things like that. So we could take a look at some questions and some clarifications. I saw that Scott had mentioned, which is thank you for that. FYI, the code allows both crucifery and Brassicaceae. Both are valid and correct. Same for the other 7 plant families with alternate names. That's really interesting that there's only 7 that have alternate names. But yeah, in the code for animals, the type genus dictates the family name, and so it's always going to be Muscady. Always Musca is the type genus of it. Now, if you found out that Musca was not actually a muscat and was something else, technically that family would be called Muscady, and whatever the other muscads were would be something else. But it gets a little complicated. And Scott, I don't know if you want to chime in on anything else about that.
He also had vicariant there, which is exactly the word that I was looking for.
Yep. Because congener just means it's in the same genus, basically. That's what I think of, at least. Okay, and then let's see. So where are catalogs found? Somebody asked. So it really depends on the group. Some people have done really good jobs of writing catalogs out, but again, it changes, so they need to be updated over years. Other groups are not cataloged at all because there's just no expert in it, and it takes a lot of time to go through all the old literature and find those names. Sometimes they also find that names have been mistaken for hundreds of years, and basically they have to kind of be the bad guys and make these changes. So. Let's see, what other questions do we have here? Just gotta scroll up a little bit further. Mike, if you see any that you want to
Someone asked, Todd asked about the Phalaenopsis question. So I had to go and look that up, but the first part of was how related are the 2 species? Well, it wasn't until they looked at, where is it? 6 different genes. So they basically, here, I'll just read this to you from a publication from last year. Taxonomic revision of the fungi from the Ceratobasidiaceae family revealed that T. basicola may form a lineage separate from the Phalaenopsis genus. So that's how it got moved to Bertone, right? A subsequent study based on sequencing 6 different gene regions not only confirmed these results, but also distinguished within this lineage 2 sister species, which were named Bertone basicola and B. ruxae. These species are considered to be cryptic, as it is not possible to distinguish them based on the morphological traits without DNA sequencing and phylogenetic analysis. So they would be just based on that, I would say, closely related. And as far as their pathogenicity, of course, now you'd have to go back and know which one you're looking at and do the pathogenicity tests, which it seems that there was B. vesicola was isolated from carrots and Lysianthus. On the other hand, B. ruxae was discovered on cotton, lettuce, peanut, hemp, and fava bean. And that particular publication talks about it on tobacco. So I don't know if that helps, but I'm thinking that a lot of the previous studies, if they didn't distinguish which one they were looking at, we may not actually have the information, as Matt mentioned, with the insects. But it's not something I've delved into until 5 minutes ago.
Thanks, Mike. And there were some questions about, are the scientific names always represented in English language? Well, the letters are technically, what is it called? I mean, they're Western script, I guess, but they're the scientific names don't necessarily have to be Latin or Greek. It's just a tradition kind of to use them. Technically, you can have native languages be used. A lot of people are naming them after what the native peoples in those regions you call those animals. They can be named after all different things. But again, there are certain rules in the codes that dictate how you can name them in certain ways. And so you do have to kind of stay within the guidelines a bit. But there is some creativity that you can have with them. And then there was a question about, which is a really interesting concept. So where was it? It was if the names change based on new understanding, why has the misnamed Magnolia host not been changed? So I don't know if that means the Magnolia should be renamed. But what happens is once a name is valid and it represents a valid species and it's the oldest name, it will stay forever. You can't really rename a species once it's a valid species that's been named once. It just, that name stays with it. So we couldn't rename that to the laurel psyllid with like Triaza lauraceae or something like that, because that would mess a lot of things up. And so we really keep the concept of priority. So the oldest name is the name that sticks. It isn't perfect, but it's just part of how we have to do it. So. All right. Any other questions? I don't know if, let's see.
Sure, we could discuss the rules all day, but we might need to
Yes.
move on to get to
Yes.
the rest of our content.
Yep. I'm
Oh,
gonna
yeah,
do.
look at the time,
I know.
as they say. Look at the
Yep.
time.
I'm gonna do some real quick bolos, and then I've got to step out to actually go to Winston-Salem to give another talk. So that's gonna be fun. But anyway, I'm here with you all now, and let's talk about some arthropod bolos. So be on the lookouts for February, late February, or March. So things you're going to start to see in March as it starts to warm up. You're going to see, especially in bare ground, you may be seeing these swarms of ground nesting bees. These are basically like little apartment complexes with multiple solitary bee homes, and they're basically harmless. They're not aggressive, and there's something that's really important to our ecosystem. So I would definitely say if you can leave them, leave them. There are ways to get rid of them otherwise, but who doesn't like these cute little bees coming out and pollinating things and being a sign of spring? You may see fall cankerworms, which are these little green inchworms with very stubby little prolegs there with the arrow. These will hang off of trees in the spring sometimes. Sometimes they defoliate. We haven't had major issues lately, but they will eat the leaves of the newly emerging leaves off of trees and bushes and things like that. You might see some termite swarmers. So these are the winged termites, the reproductive. So spring the warmth will encourage them to produce these reproductives. They'll pour out of the ground, they'll go mate. If they're pouring out of your house, call somebody, because that's not a good thing. If they're pouring out of the ground near your house or outside in the nature, it's not going to be necessarily a big major concern for you. But if you're worried about it, definitely call a professional to take a look. The paper wasps, hornets, yellow jacket queens are going to be waking up. They mated last fall and they overwintered, they hibernated, and now they're going to go out. They want to get a jump on the spring. They're going to go out, chew wood to get paper for pulp, or to get pulp to make these nests, and they're going to be active as well. And sometimes coming out inside of your house where they've hibernated. And just like those wasps, you also see a lot of larger bugs waking up, like the stink bugs and the leaf-footed bugs. You're going to see them a little bit more active and out and about. But it's still going to be fairly cool, so not a ton of activity, but things are going to start waking up. And with that, I'm going to stop sharing, and I'm going to actually have to step away. But I really enjoyed talking to you today about all these crazy scientific words and all that stuff, and hopefully you all enjoyed it too. So, all right, until next time.
Yeah, thank you, Matt, and safe travels, and enjoy your next talk.
Thanks.
That was excellent.
It's about spiders. It'll be fun. So, all right, everyone. Talk to you later.
As far as the BOLOs, be on the lookouts on the plant disease side of the equation. There are some of the same ones that we would see this time of year, not a whole lot yet, but Entomosporium leaf spot on Indian hawthorn and red tip photinia, if there are any of those left around. Something we see because these plants are evergreen, so the bright red spot's pretty typical of it. Looking closer, you see the blister-like acervuli where the spores are formed. Then another one to be on lookout for will be Sclerotinia stem rot, which could be on especially brassicas, but it can be on snapdragons, could be on a number of the herbaceous hosts, larkspur, for example. And I'll go for make a comment in a moment about how to differentiate between that and botrytis blight on, well, it can be on pansy viola, it can be on a lot of other things too. So what would we do to look for differences there? Well, first of all, the similarities. They're both cool weather pathogens. They both can cause stem rots and collapse of the shoots. Although with the botrytis, we tend to see also the leaf and flower blight. We even see canker and roses from it. They're both transmitted, or they both disseminate by airborne spores. The difference is, though, the mycelium of the Sclerotinia tends to be denser typically than the botrytis when you see it. But the sporulation is really the difference. So you don't see the gray fuzzy sporulation with Sclerotinia that you do with botrytis. The sclerotia themselves, the dense survival structures, are quite common with Sclerotinia sclerotiorum. They're loosely bound to the plant, or they can even be loose inside the hollow stems of hollow stem plants. They're less common in botrytis, but when it forms its sclerotia, they're also black, but they're more oppressed to the host tissue. Again, botrytis will sporulate profusely on the dead tissue, whereas in the case of Sclerotinia, it isn't until later when the sclerotia drop to the soil and then germinate to form very small apithecia that the spores are then produced and released into the air. Camellia petal blight, which would be a fungus caused by a fungus related to Sclerotinia, is one that we'll start to be seeing on our Camellia japonicas, and it's a good test, but not a perfect one. If you pull the base of the flower off and you see the ring of fungal mycelium there at the base, then that is your diagnosis for for Camellia petal blight. So check a couple of blossoms just to be sure in case it wasn't present in the first case. And here is a picture of, you have to really be looking for them. You can see how small it is compared to a penny, but that is the epithecium here of the fungus there coming out of the soil. That's actually right in front of your building there, Charlotte. I took several years ago. So those airborne spores will float up and infect the blossoms. Boxwood blight, something to be on the lookout for as well as usual. And our Gymnosperangium rust, the early one, of course, coming out is going to be the cedar quince rust, Gymnosperangium clavipes. It doesn't have the large gall associated with it that we see with the cedar apple rust. I'll show a photo of that in a moment, but this is one that tends to come out a little bit earlier in the season, producing the spores that will then infect the alternate hosts, such as our calorie pear. I believe it's the species that we see a lot on the calorie pears. Here is a gall of a Gymnosperangium juniperi virginiani formed on an eastern red cedar. So that'll be a little bit later, maybe April, when the first warm rains come, that we'll start seeing the big Telia horns come out of that one. And finally, to close off with turf, which is not really my area, but that would be a couple things to look out for. Would be spring dead spot, which are areas that are killed by the cold, but because the spot was infected in the fall by a fungus. So our warm season grasses like Bermuda and Zoysia, instead of greening up, they stayed dead in those spots. And fairy ring can occur on any turf, so be on the lookout for that any time of year. If you want more BOLOs, it's raining, it's snowing, you're not able to get out and garden, go and check out our website. Be on the lookouts there in the red menu bar on the left. Charlotte, I can't tell if you're talking, but it doesn't sound like we're hearing you.
Thank you. My mouse is very slow today and I'm having to do a lot of clicking. So I should be sharing screen now. Can you see my share screen with our plant these for pollinators?
Yes.
Excellent. And I did notice there was a question in the chat about what to do for Entomosporium. And just as a reminder, we don't necessarily go into the details of control in our plants, pests, and pathogens webinars. But do we have a fact sheet, Mike, on Entomosporium?
I can't think of one at the moment. And you say it's on a tree, so I'd want to know if it was on something large like a Photinia, then there's really nothing you can do if it gets so bad as you need to replace it. On a smaller, if it's on a shrub like an Indian hawthorn, there's some good resistance or some at least less susceptibility that you can choose the ones that are going to be less damaged. And I wouldn't want to go any further than that at the moment.
Yeah, but there's lots of great information out there about Entomosporium from different extension services. So I'm sure if you search online for Entomosporium control recommendations and look for recommendations from .edu sites, that information would be easy to find. So encourage people to use their search skills and use the names, the scientific names of these diseases and insects to find more information. All right, and with that, we're going to move into our new feature for this season, our Plant These for Pollinators. You may remember a couple of years ago we had Plant These instead. So we've updated, brought back our plant feature, and we're going to focus on powerhouse plants for pollinators. So what we're going to be looking at this year are plants that are readily available. So these are things you could find at a lot of nurseries and landscapes, and that are adaptable throughout North Carolina. So should be able to be things that could be grown from the mountains to the coast, with a focus on those that provide foraging and nesting habitat for pollinators, and especially our native bees. Just a general tip to maximize foraging habitat. So foraging habitat is providing the nectar and pollen that pollinators need to survive and reproduce. So to maximize that, have multiple plants in bloom every month of the growing season. So we're going to be featuring one plant each month, and hopefully that would be something that you could either add to your landscape, or maybe if you already have it, you would know, well, I've got that, so let me look for some other things. And I was so inspired, Mike, by your and Matt's talk. While you were talking, I went and looked up the meaning of our plant feature for this month. So Eastern Redbud is our Plant These for Pollinator selection for February moving into March. And the scientific name is Cercis canadensis. And I had an inkling canadensis maybe reference Canada, but I had no idea what Cercis might mean. And it is from the Greek word kerkis, which means a weaver's shuttle, and it references the shape of the seed pods. And as you can see in the picture, the seed pods, it is a legume, it's in the legume family, so they look like snow peas, actually, kind of dried up brown snow peas, so they look like beans. And this is a shuttle, a weaver shuttle. So you can see there is a strong resemblance in the overall shape. So that is where we get the name Cercis. And then canadensis of or from Canada. And you can see here the native range is part of Canada, and then much of the United States, the eastern half of the United States. So it does occur in Canada as well as all throughout the southeast, and most of the eastern United States and North Carolina. So this is the reason we typically are growing Eastern Redbud. It's the common name for Cercis canadensis, for these beautiful purple to lavender flowers that emerge early in the spring, one of our first woody plants to bloom. I anticipate it will be in bloom before our next Plants, Pests and Pathogens webinar. It often does bloom, it seems, around the middle of March to some years even earlier March. This year, I think it's going to be probably a little bit later than it has in some years. I think we've had a little bit of a colder winter. But it is very popular as a small deciduous flowering tree for landscapes. It is, of course, if we already saw, it's native to the eastern U.S. and throughout North Carolina. It is relatively fast growing, which is one thing that also helps make it popular, and ultimately can grow to 20 to 30 feet, although there's a lot of range with the varieties and the named cultivars. Some of those can be more compact. And it tends to be similar in width to height, so it's kind of a wider spreading tree, or kind of that overall shape is a little bit of a spreading tree. So make sure you have enough room when you do place it in your landscape for it to grow tall and wide. It's very adaptable. It will tolerate a range of soils, really anything other than extremely wet and extremely dry, but it's going to thrive in well-drained soil and sun to part shade. It also tolerates a wide range of pH. So whether you have a higher pH or a lower pH, it's pretty adaptable there. The one thing you do need to look out for are deer. They love it. So young trees, especially that are in the range of deer feeding, you'll want to protect them. And you can do that, with some wire fencing kind of around it. It can be very effective for protecting them until they get up tall enough to not be right there at mouth level for deer. And these beautiful purple flowers line the stems and even come out on the larger trunks. It's quite amazing, the profuse flower production of our Eastern Redbud. And as I mentioned, usually March and into April before the leaves come out. And they often will still have some blooms on them as the leaves come out. And besides being pretty and just really a harbinger of spring, these flowers are a very important early nectar source for honeybees and native bees. It is at these kind of ends of the seasons, the early part and the late part, that it's very important. We make sure that there's some things out there for bees to forage and pollinators as they start to wake up from their winter hibernation and are active on these warmer days. There's lots of stuff typically out there in the middle of the growing season, but early and late is when we can do a lot of good by providing some things to make sure there are resources there for our pollinators. Okay. And one thing I always love about redbud that you often see are on the edges of the leaves, you see these almost perfect little circles have been cut out. This is usually a little bit more into the growing season, later in spring and in the summer. And this is the activity of leafcutter bees. And we have a couple of species in the Megachilae, I hope I said that correctly, genus of these leafcutter bees here in North Carolina. And they do favor redbud. I think it's because of the texture of the leaf. There's other things, you'll often see them doing this on things like roses and muscadine grapes. And there's some plants that I think just because of the texture of the leaf are particular favorites of theirs. But it's very noticeable on redbuds because they have those large heart-shaped leaves and it just looks like scalloping around the edge. So these leafcutter bees cut these little circles out and they take it back to their nest, which is, they're just solitary bees. So they are usually nesting in holes in wood or perhaps bee hotels. And they line the nest with these little circles and they put a ball of pollen in there and they lay an egg. And then that is going to be a baby bee that will develop this year and hatch out next year. So this is just a picture of kind of that bee nest that would be inside one of these hollowed little holes or stems or somewhere that the bee is found to nest. And this picture actually comes from my days as an extension agent, actually when I was in Pender County, and somebody had emailed it to me and said, every time I turn on my outdoor spigot, all these little pieces of leaves come out. And it was just, they could not figure out how in the world or why in the world the leaves were in there. The person thought that maybe kids were doing it. And I strongly suspect it was leafcutter bees, that they liked the size of the spigot, the opening, and they thought, well, this would be a great place to make a nest. And so they were putting in the little pieces of leaf they'd cut, but each time the person turned the spigot on, they would come back out. So hopefully that bee got the message and moved on and found another appropriate place. That's just an interesting picture resulting from their activity. All right, my mouse, there we go. When you go look for redbuds at a garden center, you are likely to find probably some of the name varieties. And most of these name varieties have been selected more for their foliage than their flowers. Most of them still have the very prolific lavender purple flowers. There are a few varieties out there that have maybe white flowers or pinker colored flowers. But really in the last, say 10, 20 years, most of the focus has been on varieties with different colored foliage or a weeping habit. So NC State has actually had a breeding program, and these are some of their releases that you could potentially find at garden centers. And they kind of represent some of the types of varieties that are out there. So purple leafed varieties, this one is named Merlot, and it's kind of a compact one because it's actually crossed with the subspecies of Cercis canadensis, the subspecies texensis, which occurs more in Texas, which is a more compact tree. So very purple foliage, compact and upright. There's several purple leaf varieties out there. Some of them will kind of go towards green in the summer. The leaf color is most intense in the spring when it first comes out. Then there are varieties with golden colored foliage and weeping habit. So there's some upright golden leaf ones. This Golden Falls, another NC State variety, is a weeping golden leaf variety. So you can see there, it just has this very weeping, almost trailing habit. and these bright gold leaves. Again, with the gold leaf varieties, the color's most intense in the spring, and they go a little bit more towards regular green in the summer. And a new one that is very popular is Flamethrower, and it kind of has both. It has the golden foliage, and then the newest leaves are more intensely colored with this kind of red to burgundy colors. And it is more of an upright tree, that kind of normal size, kind of wide spreading habit. So you want to make sure you have room for it when you place it in the landscape. But you'll get this really intense color on the new growth, especially in the spring. And even when it throws out new growth in the summer, those leaves can be intensely colored, and then it shades back to yellow and green more on the interior of the tree. So lots of varieties out there. And if you want to learn more about Redbud, and I will also mention anything that was the links to the names of the plants that we'll be talking about during this feature will always link to the plant toolbox. So you can find and learn all about Cercis canadensis, including how to pronounce it. So one thing that you will see in the plant toolbox for each plant profile is that little icon. It's like a speaker. If you press that, it'll tell you how to pronounce the name. So I encourage you to learn more about Cercis canadensis. There's lots of great pictures. There's things that could be confused with when you're trying to identify it. There's actually some lists of alternatives. Lots of wonderful information there about our Eastern Redbud and
Charlotte, if
other plants. Yes.
if I can just interject two things really quickly. One is now, well, the name I was just checking appears to go actually back to Linnaeus. So this is an L after it, and I got that from the International Plant Name Index, which I'll put that in the chat real quick. IPNI.org is where you can look up the plant names. We gave I gave the fungal one, and Matt mentioned the insect ones and arthropod ones. The other thing I would be remiss in not mentioning is that there has been a lot of problems with a disease. We're calling it a disease. It's not actually been proven to be a disease, vascular streak dieback. It is affecting a large number of woody hosts, but far and away the most common one is our Cercis canadensis. So I would, if I were going to be buying a Redbud, I would be sure not to buy it dormant so that I can evaluate well that there's no dieback going on, that all the leaves appear to be normal and so forth.
Thank you. Is that disease mostly occurring in nurseries, or are you seeing it in established plants in the landscape? Like it didn't come from, it didn't come in with the plant?
Yeah, it's mostly, from what I've seen, it's mostly been in nurseries or in plants recently transplanted from the nursery. That's not to say it couldn't get into plantings that are already established. I just can't say I've seen that yet. Yeah, there's evidence of that happening, circumstantial anyway, for other hosts. I don't remember if there is. I think there is some circumstantial evidence in Redbud as well. So the short answer to your question is, my personal experience, it's been nurseries and recently transplanted material, but don't rule it out.
So you said it's not certain if it is actually a pathogen, so I guess they're not sure how it's spreading.
Well, we assume it's spreading by basidiospores in the air, but because partly because the fungus is so hard to culture, the official steps called Koch's postulates haven't been completed yet to actually say that it has been proven to be the cause. But it's I don't really have any doubt about it myself. I see that someone has put in a link to a fact sheet. There's also a new fact sheet that has just come out about it. So I can actually, if there's, we only have a minute left, but there is a new fact sheet that just came out about vascular streak dieback. Let's see. Okay, here it is. I'll drop that in the chat as well. I would not, there's some mention of chemical products there. I wouldn't go that route, but there's some other good information that if you want to check it out.
All right, thank you. I'll be sure to capture those for our resource document as well. And I also noticed a couple of people were commenting on the the foliage color, and that can potentially affect leaf feeding insects, so caterpillars. I did a really quick search, but not intense search, on whether anybody had looked at these varieties with different colored leaves as to whether it has negatively impacted leaf feeding insects or reduced leaf feeding by insects, and I didn't see anything. But again, I didn't go super in depth. But in general, the anthocyanins that cause the purple foliage can deter or change the feeding habits of things like caterpillars that would rely on that foliage. I don't know whether the leafcutter bees, whether it would affect their behavior or preference for those purple leaf varieties. But these varieties still do produce tons of flowers, and the same time, the same kind of color as the native species. So I wouldn't expect there to be a lot of different activity by pollinators actually visiting the flowers for nectar and pollen. So really it would be more whether the foliage is available there as a resource for other insects. All right. So
And as long as you're talking about flowers, I don't know if anybody else noticed, but I did this morning. It looks like the pollen has started.
yes, I have noticed pollen starting too. And things like red maple are blooming, and winged elm. Around my house, we have the American filbert. They're blooming. So yes, pollen is getting started, and I could feel it in my throat as well. While we were mentioning Redbud, I did want to mention a citizen science opportunity. So there is a project to track Redbud development. This is across the United States, as just a way to observe seasonal change and how things change from one season to the other. So if you have Redbuds in your landscape or your community and you'd like to be part of this Redbud phenology project, you can find more about it by just searching Redbud Phenology Project. And of course, we'll send the link out with the resources and the recording. But this is a neat project that anybody could be part of. And we'll close and with just a few announcements. I know we're running a little bit long, but lots of great stuff today. And one thing I want to let you guys know about, and we'll be sending more information about in the next week or so, is a continuing education webinar. That will be the day after our next Plants, Pests, and Pathogens webinar. It'll be on March 26th, and it'll be about building a better monarch butterfly garden. We'll hear from Dr. Dan Potter, who is an emeritus professor with the University of Kentucky and has done a lot of research on planting for monarch butterflies particularly, and on a home landscape scale. So we'll learn about the optimal garden design, the plant species, and best practices to sustain healthy monarch populations, or do all that we can do in our landscapes. So this is sponsored by the North Carolina Extension Master Gardener Endowment, and we will be having registration posted on the EMG Internet very soon. So I hope you'll join us then. It also coincides with Day of Giving, which is NC State's annual Day of Giving, where we encourage people to support things they care about. And with the NC State Extension Master Gardener Endowment, we actually are very excited to say we have a donor, an anonymous donor, who has pledged a $10,000 challenge gift. So every dollar that is donated to the NC State Extension Master Gardener Endowment on Day of Giving, specifically on Day of Giving, they will match 3 to 1. So if we can get a little over $3,000 in donations, our donor will give $10,000. So we'll end up with $13,000 more in our endowment to support things like continuing education for Extension Master Gardener volunteers. For example, we were able to offer scholarships to EMG College last year through the endowment. So you'll be hearing more about that as well. You've probably also seen emails about the Gardens, Lawns, and Landscapes, the online courses that Scott Zona is now the course instructor and will be leading throughout this year. There are a series of 5 classes, each is 6 weeks long. They are asynchronous, which means you participate on your own time within the time frame of the course. There is a discount for Master Gardener volunteers and extension professionals, and you can learn that more about the courses on the Extension Gardener website. If you have not yet heard, I encourage you to save the date for the International Master Gardener Conference coming up August 25th through the 28th. This will be online this year, so it'll be hosted by the Texas Master Gardener Program, and it'll be a virtual conference. So they really look forward to making the conference accessible to people all over the United States and really around the world by hosting it as a virtual conference. And they have lots of wonderful sessions planned. You can learn more about it online on their website, and they are anticipating that registration will open in April. Another thing, if you are familiar with the International Master Gardener Conference, you know that it is always also includes a Search for Excellence Awards program for the Master Gardener program across the United States and in other countries around the world that have Extension Master Gardener program. So part of the conference this year will be the International Master Gardener Search for Excellence. This is held every other year, and North Carolina has a strong history of doing very well and having received several awards over the past years. You can see in the picture here on the left, our Master Gardener volunteer and former agent from Guilford County accepting an award. I think this was for a demonstration garden. And on the right, we have Master Gardener volunteers and the current agent there, Ashley Troth, accepting awards for Durham County in demonstration gardens and innovative projects. So I encourage you all to think about the work you're doing in your counties and take a look at the Search for Excellence application process and think about getting your applications in by the deadline, which is May 1st. So you have a couple of months. We'd love to see some of our projects and Master Gardener volunteers from North Carolina recognized. And that is it. We appreciate you joining us today. Next month, we're really excited to welcome Amy Bruzacchese, who is the Extension Therapeutic Horticulture Programming Manager. She's here at NC State in the Department of Horticultural Science. She will be talking about From Hobby to Healing, Understanding Garden Groups and Therapeutic Horticulture. That wraps up our webinar for today. And we will be sending out the recording and posting it on our EMG intranet. Until next time, I hope everybody has a wonderful gardening season, a wonderful start to spring, and we look forward to seeing you in March.